Somewhere in a research facility in Novosibirsk in 2012, a bone fragment barely 2.5 centimeters long sat inside a storage drawer, indistinguishable from roughly 2,000 other scraps of bone stacked around it. For 4 years, it carried a single word on its label: unidentifiable. It had come out of Denisova cave, tucked into the Altai mountains of southern Siberia, a cave that at different points in prehistory had sheltered Neanderthals, Denisovans, and modern humans, sometimes within the very same stretch of stone. Nobody in that room in 2012 had any reason to think this particular fragment was different from the rest of the pile. That changed in 2016, when a team led by Oxford researcher Tom Hyam began running a technique called ZooMS, a method that fingerprints collagen proteins well enough to sort bone by species, even when the bone itself gives nothing away to the naked eye. This fragment came back flagged as human. But which kind of human, nobody could yet say. The answer took 2 more years to arrive. Researchers at the Max Planck Institute, led by Viviane Slon and Svante Pabo, sequenced its full genome, and the results were published in Nature in August 2018. The bone belonged to a girl, at least 13 years old, who had lived roughly 90,000 years earlier, and she was not one species. She was 2 at once. So what exactly was sitting inside that drawer for 4 unremarkable years? All right, if ancient bones casually rewriting the species tree is your thing, stick around, because this one only gets stranger. Hit subscribe if you want more of this. Her name, once scientists worked it out, was Denny. And Denny's genome didn't just surprise them. It broke the model they'd been working from for a decade. Denisova Cave was, at various points, home to Neanderthals, Denisovans, and modern humans, the only known site on Earth where all 3 overlapped. A Neanderthal toe bone, pulled from layer 11.4 in the East Gallery and found in 2010, dates to the same window as the Denisovan finger bone that first put this cave on the map. And yet Denisovans left behind almost nothing physical to study: a handful of teeth, that one finger bone, and the long bone fragment that would turn out to be Denny. Stone tools scattered through the region, mostly Mousterian in style, are linked to Neanderthal activity and dated to around 40,000 years ago. The cave's own layers stretch across more than 280,000 years of shifting, overlapping occupation, one group moving in as another moved out, or, in some cases, staying long enough to cross paths. When modern humans eventually arrived in this part of Siberia, the layering suggests they missed some of these earlier populations by a matter of thousands of years. Not by tens of thousands, not by whole geological ages. None of that explained how a Neanderthal and a Denisovan ended up raising a child together. That answer didn't come from the cave, the tools, or the layers. It came from the bone itself. Denny's fragment held enough intact genetic material for a full nuclear genome reconstruction, a level of preservation that surprised the researchers working with material this old. The sequencing team ran her genome against reference genomes already pulled from other Denisova cave specimens, one Neanderthal, one Denisovan, and started comparing. Roughly half of Denny's genome matched Neanderthal markers, the other half matched Denisovan markers, an even 50-50 split right down the middle. That ratio isn't what you'd expect from someone several generations removed from a mixing event, with archaic ancestry diluted down through a long chain of descendants. It's the signature of something much more direct, a first-generation offspring, one parent from each side. Before Denny, every claim of interbreeding between these groups had come from indirect evidence, small residual percentages of archaic DNA turning up in the genomes of living populations, a kind of genetic echo. Denny gave researchers something they'd never had in hand before. Not an echo of an old encounter passed down and diluted, an actual child standing as direct proof that the encounter happened. Once the team looked closer at exactly which Neanderthal and exactly which Denisovan had produced her, the story took a second turn nobody in the lab had been expecting. Denny's mother's DNA turned out to be more closely related to Neanderthals recovered from Vindija Cave in Croatia, thousands of kilometers to the west, than to the Neanderthals whose remains had been found in Denisova Cave itself. That single detail carries a lot of weight. It tells you Neanderthal populations were not sitting still in one place. Her mother's lineage had migrated across Eurasia at some point, after roughly 120,000 years ago, covering enormous distances for a population without maps, roads, or any of the tools modern humans associate with migration. Her father was different, a local Altai Denisovan, closely matching later Denisovan specimens pulled from that same cave. But his genome held its own surprise. Buried inside it was an older, separate trace of Neanderthal ancestry, from an entirely different mixing event that had happened generations before Denny was ever born. Put plainly, Denny's family tree contains at least two independent instances of Neanderthal-Denisovan interbreeding, not one. The scientists behind the 2018 Nature paper stated this directly. Finding a pattern like this suggested that whenever these two groups crossed paths, mixing between them was common. Not a rare accident, caught once by pure luck in a single bone fragment. A repeatable pattern, showing up twice over, inside the ancestry of one girl alone. If two different species could interbreed this easily in one Siberian cave, the next question is how far that habit actually spread beyond it. Denisovans exist, as far as science is concerned, almost entirely because of a single finger bone sequenced back in 2010. No skull, no skeleton, no agreed-upon physical description of what one of these people would have even looked like standing in front of you. They hold the distinction of being the first hominin group in human history, identified purely through genetics, not through the shape of a fossil. Only 5 specimens from Denisova cave have ever been formally attributed to them, along with a scattering of teeth and partial bone fragments. That is the entire physical record of an entire species. And yet their genetic fingerprint doesn't stay confined to that one cave. It shows up today in the genomes of populations thousands of miles away, in Papua New Guinea, in Australia, in the Philippines, in Tibet. That kind of spread implies Denisovans once ranged across a vast stretch of Asia, far beyond the narrow mountain valley where their remains happen to survive. Scientists still can't tell you what they looked like. The entire identity of a vanished species rests on DNA fragments smaller than a fingernail, pieced together from a handful of bones that could fit in the palm of one hand. One place that DNA turned up would go on to change a much bigger story, 3,280 meters up on the roof of the world. The Xiahia mandible is a fossil jawbone that surfaced almost by accident, pulled from Baixia Karst cave on the Tibetan plateau back in 1980. For nearly 40 years it sat in storage, unidentified, just another piece of bone waiting for the right technology to catch up with it. That technology arrived in 2019, when a team led by Fahu Chen and Dongju Zhang ran ancient protein analysis on the fragment and identified it as Denisovan, dating it to at least 160,000 years old. That makes it the earliest known hominin fossil ever found on the Tibetan plateau, older than the oldest known Paleolithic sites in the entire region by more than 120,000 years. The implication is direct. Denisovans had already adapted to one of the harshest, most oxygen-starved environments on the planet, tens of thousands of years before Homo sapiens ever set foot up there. Surviving at 3,280 meters isn't a small feat. The human body's default response to that kind of altitude is to overproduce red blood cells, and for most lowlanders, that response becomes dangerous rather than helpful. Living there long-term requires suppressing that reaction entirely, a specific physiological trick most people simply don't carry. Present-day Tibetans and Sherpas do carry it, in the form of a gene called EPAS1. That gene didn't evolve inside modern humans at all, it was borrowed. EPAS1 governs the body's hypoxia response, the entire chain of reactions that determines how the body copes with low oxygen. Tibetans carry a version of this gene that blunts the usual overproduction of hemoglobin at altitude, the same overproduction that causes cardiovascular strain and pregnancy complications in most other populations trying to live that high up. When researchers went looking for where this exact version of the gene came from, they found it sitting in Denisovan DNA, and almost nowhere else on the planet, aside from Tibetans themselves, and a low frequency of it in Han Chinese populations. Oceanian populations carrying heavy Denisovan ancestry, Papuans among them, don't have this variant at all, simply because their environment never demanded it. That's adaptive introgression in its purest form. A single useful gene crossing the boundary between two separate species, then getting selected for on its own, completely independent of whatever else came along with it. In effect, an extinct hominin handed modern humans a mountain survival tool it had already spent tens of thousands of years perfecting, before Homo sapiens ever needed it. Move southeast, off the plateau and down into the Pacific Islands, and the Denisovan signal doesn't fade out, it gets stronger. Papuan Highlanders and Aboriginal Australians carry roughly 4 to 6% Denisovan ancestry, a figure that stood for decades as the highest documented anywhere on the planet. Compare that to the roughly 1 to 3% Neanderthal contribution found across most non-African populations worldwide, and the scale of it becomes obvious. Denisova Cave sits in Siberia. Papua New Guinea sits more than 6,000 kilometers away. That distance alone raises an obvious problem. How a population without ships, without agriculture, without any of the infrastructure modern humans take for granted, managed to spread its genetic signature across that kind of range. Genetic modeling offers part of an answer, pointing to at least two separate Denisovan lineages contributing to different modern populations, which implies these hominins weren't confined to one mountain range in Siberia at all, but held a large and geographically dispersed territory. Some researchers point to 200,000-year-old stone tools recovered on the Indonesian island of Sulawesi as a possible, though still unconfirmed, trace of that spread, evidence that something capable of shaping stone was moving through the islands long before modern humans arrived. For years, Papua New Guinea Highlanders held the record for the highest Denisovan ancestry found in any living population on Earth. That record stood until 2021, when one study looked in a place nobody expected to find anything at all. That record didn't just get broken. It got broken by a group living hundreds of kilometers away, in a country most people wouldn't think to associate with archaic DNA in the first place. In 2021, a study published in Current Biology, led by Maximilian Lorena and Matthias Jakobsen of Uppsala University, analyzed 2.3 million genotypes drawn from 118 different Philippine ethnic groups. Buried inside that dataset was a Negrito group called the Aita Magbukon, concentrated in the province of Bataan. The Aita Magbukon were found to carry the highest Denisovan ancestry ever recorded in a living population, close to 5% of their entire genome. That works out to roughly 30 to 40% more Denisovan DNA than either Papua New Guinea Highlanders or Aboriginal Australians, the two groups that had held that record for years beforehand. Part of the reason the signal stayed this strong comes down to timing. The Aita Magbukon experienced less recent East Asian admixture than most other Philippine ethnic groups, and less dilution from newer ancestry meant more of the original archaic signal survived intact into the present day. Researchers concluded this wasn't simply an extension of the same event that gave Papuans their Denisovan ancestry. It reflected an entirely independent admixture event, Denisovans mixing separately into the ancestors of Philippine Negritos, on a different timeline, likely through a different route altogether. The country holding the world record for Denisovan ancestry isn't Papua New Guinea, it isn't Australia, it's the Philippines. And the Philippines wasn't only home to interbreeding with Denisovans. It may have been home to an entirely separate archaic species of its own. Homo luzonensis is a small-bodied archaic hominin, formally described from fossils recovered on Luzon Island in the Philippines. The researchers behind the 2021 Denisovan ancestry study drew a direct line between the two findings, suggesting that multiple archaic groups inhabited these islands before modern humans ever arrived, and that these groups may have been genetically related to one another. Put plainly, that means the Philippines potentially hosted at least two distinct archaic hominin populations, layered on top of each other across time, long before any Homo sapiens set foot on the islands at all. Island Southeast Asia doesn't offer the same advantages Siberia or Europe do for this kind of research. There are almost no cold, dry caves in the region suited to long-term DNA preservation, which means most of this history has to be reconstructed from the genomes of living people rather than pulled directly from fossils in the ground. That gap between what's preserved and what actually happened matters. It means the true number of archaic populations that once lived across these islands could be considerably higher than what's currently confirmed by hard evidence. Every additional case that does get confirmed adds more pressure to the old, tidy picture of a single wave of modern humans arriving and simply replacing everyone who came before them. Head back to mainland Eurasia, and that same replacement story falls apart again, in an entirely different way this time, one written into a single skeleton pulled out of the ground in Romania. Oasis 1 is a modern human jawbone, somewhere between 37 and 42,000 years old, pulled out of Peștera cu Oasis in Romania. In 2015, a study published in Nature, led by Qiaomei Fu, working alongside David Reich and Svante Pääbo's team, sequenced his DNA in full. The results stunned the field. Somewhere between 6 and 9% of his genome came from Neanderthals, the highest figure ever measured in a modern human, roughly triple the typical 1 to 3% found in living Eurasians today. This wasn't a small statistical bump, it was a massive, unmistakable signal. 3 of his chromosomal segments carrying Neanderthal ancestry were long enough, over 50 centimorgans each, for researchers to calculate exactly how recently that ancestry had entered his bloodline. The math came back specific. A Neanderthal ancestor sitting just 4 to 6 generations back in his family tree, meaning under 200 years before he was born. And yet, despite carrying that much Neanderthal DNA, Oasis 1 doesn't share unusually high ancestry with later Europeans. His specific population line appears not to have survived in any meaningful way into the people who came after him. A Neanderthal great-great-grandparent in a man who lived 40,000 years ago. On a genetic timescale, that's practically living memory. His story tells you this wasn't a single, isolated meeting somewhere in the Middle East 60,000 years ago. It kept happening, in different places, for tens of thousands of years afterward. The standard model has long held that Neanderthal ancestry in non-African populations sits at around 1 to 3%, traced back to a single mixing episode in the Middle East roughly 50 to 60,000 years ago, a figure built from the original 2010 draft Neanderthal genome. Oasis 1 shows that framework was incomplete. Interbreeding also happened locally, inside Europe itself, continuing right up until Neanderthals were disappearing from the region entirely. Neanderthals are believed to have vanished from Europe somewhere between 39 and 41,000 years ago, placing Oasis 1's lifetime right at the edge of that extinction window. The timing suggests some of the very last Neanderthals in Europe were still having children with the modern humans arriving there, rather than simply being pushed out or replaced by them. That detail complicates the old idea of a clean, total replacement of Neanderthals by modern humans sweeping across the continent. It also suggests the fossil record likely still holds other Oasis-style surprises, sitting unexamined in bone fragments nobody has thought to test yet. It wasn't only Europe and Asia where this kind of hidden ancestry was waiting to be uncovered. It turned up somewhere the textbooks had insisted it simply could not exist. For years, the standard model held that Neanderthal and Denisovan ancestry existed exclusively outside Africa, since neither species was believed to have ever lived there. In 2020, Arun Durvasula and Sriram Sankararaman of UCLA, publishing in Science Advances, compared 405 whole genomes drawn from 4 West African populations against reference genomes from both Neanderthals and Denisovans. What they found didn't match either species. It pointed instead to a separate, previously unknown archaic population, one science had never identified or named before. Statistical modeling estimated these West African populations derive somewhere between 2 and 19% of their ancestry from this unidentified archaic source. The researchers traced this ghost population split from the ancestor of Neanderthals and modern humans to somewhere between 360,000 and just over 1 million years ago. The actual introgression, the point where this population's DNA entered the ancestors of living West Africans, is estimated to have occurred sometime between the present and 124,000 years ago, a window wide enough to suggest the mixing could have been surprisingly recent. West Africans were long treated as the one population untouched by archaic mixture, the control group in every comparison. Instead, they carry evidence of an entire archaic species that science hasn't even managed to name yet. The researchers found something else buried inside that ghost DNA. Genes that didn't just quietly survive the intervening millennia. They spread. That same study identified 33 gene loci in the Yoruba population and 37 in the Mende population, where this archaic-derived DNA sits at frequencies above 50%, an unusually high rate for genetic material inherited from an extinct, unrelated lineage. One of the shared high-frequency genes is NF1, a tumor suppressor gene, which suggests this ancient DNA wasn't simply neutral leftover material. It may have been actively favored by natural selection over tens of thousands of years. The researchers themselves noted real uncertainty here. It remains unclear whether this reflects a single interbreeding event or several separate ones, and whether just one archaic population was involved, or multiple distinct ones layered on top of each other. No fossil, no skeleton, no physical remains of any kind have ever been linked to this ghost population. It exists entirely as a statistical signature embedded inside the genomes of living people. Put plainly, an entire hominin lineage may be genetically documented in detail while remaining, physically, completely unknown to science. It stands as the clearest case yet of a species whose only surviving evidence is other people's DNA. As unsettling as an undiscovered species hiding inside living genomes sounds, an even older ghost was waiting one layer further back. In 2020, population geneticist Alan Rogers and his colleagues, publishing in Science Advances, modeled genetic patterns across Neanderthal, Denisovan, European, and Asian genomes together. Their analysis pointed to a population that had split off from the rest of the human family tree, somewhere between put it all together, and the picture that emerges is layered. Modern humans mixed with Neanderthals. Modern humans mixed with Denisovans. Neanderthals and Denisovans mixed with each other. an unnamed ghost population mixed into the ancestors of West Africans, and a super archaic lineage mixed into both Neanderthals and Denisovans, separately, at different points in time. That's a minimum of 5 distinct interbreeding events across the hominin family tree, all documented through genetics alone, with strong reason to think more remain undetected. Scientists like Murray Cox at Massey University have described this pattern plainly. Whenever different hominin groups crossed paths, interbreeding was the rule, not the exception. That directly contradicts the older, tidier model most people grew up with, the idea of a single, pure lineage of modern humans sweeping across the planet and simply replacing every other species it encountered. The evidence points somewhere else entirely. The modern human genome behaves more like an archive than a clean bloodline, built from layers of DNA belonging to populations that no longer exist as distinct species but never fully vanished either. Every population studied so far carries its own unique combination of these layers. Tibetans carry one combination, Melanesians carry another, Filipinos, West Africans, Europeans. Each population holds a slightly different archive, shaped by its own specific history of contact. Denny, the girl found in that Siberian storage drawer, was never an exception to this pattern. She was proof that it was happening constantly, everywhere researchers finally knew to look for it. The 2018 Nature paper on Denny reached a conclusion worth sitting with. Finding a first-generation hybrid among the small number of archaic specimens sequenced to date suggested that mixing was common whenever these groups crossed paths, not some rare one-off event. Consider the odds embedded in that statement. Fewer than a dozen high-quality archaic genomes have ever been fully sequenced in the history of this field, and one of that small handful turned out to be a direct hybrid child. Statistically, that's an extraordinarily high hit rate if hybrids were actually as uncommon as scientists once assumed. Every new technique applied to this question has only uncovered more mixing, never less. ZooMS protein screening turned up Denny. Ancient protein analysis identified the Shahi mandible. Statistical ghost population modeling revealed the archaic signal buried in West African genomes. None of these 5 interbreeding events described across this investigation were predicted in advance. Each one was found by accident, sitting inside a fragment or a genome that nobody expected to hold anything unusual at all. That track record suggests the current list of interbreeding events isn't a ceiling, it's a floor, which raises an uncomfortable question about all of this evidence. How much of it was actually found on purpose? Only one first-generation hybrid fossil has ever been confirmed across the entire hominin fossil record, spanning hundreds of thousands of years and multiple continents, and that fossil is Denny. The ghost population identified in West African genomes has no known fossil at all connected to it. The super-archaic lineage has no confirmed fossil either, existing only as a statistical shadow embedded inside later species DNA. Vast regions of the planet where archaic populations likely once lived, much of Africa, islands Southeast Asia, Central Asia, have barely been sampled for ancient DNA at all, largely because warm, humid climates destroy genetic material long before anyone gets the chance to study it. Every confirmed case in this entire investigation came from a small number of well-preserved cold climate sites, which means the record researchers are working from is almost certainly biased toward whatever happened to survive, not toward what actually occurred across the species' full range. Newer sequencing methods keep finding signals in places nobody was specifically looking for them, from a storage drawer in Siberia to a population survey conducted across the Philippines. If a single overlooked bone fragment sitting in a drawer was enough to prove an entire species had children together, the only real question left is what else is still sitting, unidentified, in a box somewhere, waiting for someone to finally run the test. That question is exactly why this search hasn't stopped.